This is part 3 on the cell division lectures. We left off with this slide on anaphase. And this is the phase of mitosis or karyokinesis, where the sister chromatids separate at the centromere, which is just this area where they're connected. And the two regions where they're connected are called the kinetochores. Each sister chromatid is now called a daughter chromosome, and it's pulled rapidly toward the centrosome, which means opposite ends of the parent cell. The way that happens is the spindle fibers or spindle microtubules are attached to each of those daughter chromosomes, and then they shorten. Another thing that happens is the cell becomes elongated or oval shaped. During telophase, the daughter chromosomes have reached opposite poles of the cell. And remember I told you telophase was opposite of prophase. So if in prophase the chromatin condenses and forms chromosomes, now the chromosomes are going to decondense or unravel back into their original form called chromatin. The mitotic spindles and the spindle fibers will be dissolved. The nuclear envelopes will reform around the daughter chromosomes, and the nucleolus will reappear. And these are all opposite of the events that we learned in prophase. In cytokinesis, it depends on what type of cell it is as far as how cytokinesis occurs. In plant cells, it is different than in an animal cell. In a plant cell, the Golgi apparatus forms vesicles in between at the very middle of the parent cell, and those vesicles eventually form into a cell plate, and that cell plate eventually forms into a cell wall, and you have two new cells with both surrounded by a cell membrane and a cell wall. In animal cells, the original parent cell forms a cleavage furrow, which is what you see here. And then those Golgi vesicles form a contractile ring, which pinches the parent cell in two. And now you've got two daughter cells here and here that are clones of the parent. Now, it is very important that the cell cycle be regulated because, as we said in the beginning, in the first lecture, if the cell cycle goes unregulated, you can have things happening in the human body as minor as psoriasis and as major as cancer. So the cell cycle has to be regulated, and external factors will initiate cell division. One thing that can initiate cell division is the death of a nearby cell. So when a cell that's near to another cell dies, it can cause the cell that doesn't die to divide, to replace the one that died. Another thing that can initiate cell division is the release of growth promoting hormones, such as, I'm sure you've probably heard of this one, HGH, human growth hormone, and the cell size. As a cell grows, it's only going to grow so big. If you remember learning this, I think it was chapter 4, where the cell is small because that increases its surface area compared to its volume. So dividing will increase that surface area to volume ratio. So I'll write it like that. Dividing will increase the surface area to volume ratio. And that will make a cell more healthier. And cells that are crowded together can inhibit cell division because obviously more cells is just going to make more crowding. So those are things that can control the cell cycle. Those are external things. Now there are internal controls as well, like chemicals. The cell cycle is controlled at 3 checkpoints. There's a G1 checkpoint that checks the integrity of the DNA. There is a G2 checkpoint that checks whether or not the DNA replicated properly, proper chromosome duplication. There is an M checkpoint that determines if each spindle fiber has attached to each kinetochore of the kinetochore of the sister chromatid, this part right here. The kinetochores are located in this region. If each spindle fiber has attached there. Of course, that would be in prophase. That would happen in prophase before metaphase. So this is a pie graph of the checkpoints in the cell cycle. Starting with, we will start with the G1 checkpoint. So you have interphase. G1, we learned what happened in G1, that the cell grows. Some of the organelles will be duplicated at this point. Some of the nucleotides needed for the replication of DNA, some of the histone proteins will be produced. Anything necessary for DNA replication is going to be produced during the G1 checkpoint. And if those things don't happen, then the cell division will stop at G1. It will be ended at the G1 checkpoint. In other words, the cell will not continue to divide. Well, but if everything checks out, if everything's okay, the S stage will get started and it will continue all the way to the DNA will be replicated, and then the G2 stage will begin. During this stage, the cell may grow a little bit more and it may produce some of the proteins necessary for cell division, like the spindle microtubules. But if something happens and at that G2 checkpoint everything's not the way it should be, then the mitosis will not occur. Okay, so cell division will be stopped here. And then there's the M checkpoint that checks to make sure that the kinetochores are attached to the spindle fibers, and if they're not, cell division will be stopped at that point and the cell won't divide. There are two groups of proteins called cyclins and cyclin-dependent kinases. Now you know if it has ACE on the end, it's an enzyme. So cyclin-dependent kinases are enzymes. But this combination of cyclins and cyclin-dependent kinases, the levels of these proteins during different phases are responsible of the progression of the cell through the checkpoints. So for example, after the G1 phase, when you get to the S phase, the level of cyclin E should be here. It should be at a peak, and the level of cyclin D should be increasing, but not at its peak. When you get all the way to the end of the G2 phase, the levels of cyclin A and D should be about the same. Sorry, didn't mean to do that. And the level of cyclin B should be almost at its peak. So when these levels are not at the levels that they are supposed to be, then that causes the cell to stop at that point of cell division and not go on with cell division. So this is the combination of cyclin-dependent kinases and kind of how they work. Cyclin binds to CDK, cyclin-dependent kinase, and then a phosphate donating protein called kinase donates a phosphate to the CDK. And then that phosphate is added to a target protein. In other words, the activated CDK cyclin complex phosphorylates the target protein. All that means is that the CDK adds a phosphate to a target protein. And now that the protein is phosphorylated, the cell cycle can advance to the next stage. But if this phosphorylation doesn't occur, then that means something is wrong, and the cell will stop at that point of cell division. There are also negative regulator molecules that can halt the cell cycle until specific requirements are met. So retinoblastoma proteins are a group of tumor-suppressing proteins. They're called retinoblastoma proteins. They are tumor-suppressing proteins that are common in many cells, which act primarily at the G1 checkpoint, which means that at the G1 checkpoint, if the cell's not ready or has not met certain conditions, it won't go on to the S stage where DNA is duplicated. Molecules P53 and P21 prevent the duplication of faulty DNA. And that can be various things. I mean, it can be that the DNA is missing some genes, or is some of the sequences of nucleotides are incorrect, but in some way the DNA is faulty. Then RB, which is on the next slide, will halt the cell cycle and release its hold in response to cell growth. Okay, so the RB regulation of the cell cycle. The RB is your target protein. Okay, and it says unphosphorylated RB binds to transcription factor E2F. So this is a transcription factor, and this is a protein named RB. And here's your DNA molecule, of course. It says E2F cannot bind the DNA, and transcription is blocked at this point. Then, as the cell grows, ATP is broken down, releasing a phosphate, and that phosphate is added to the RB protein, which releases the E2F transcription factor, and when it binds to the DNA can now copy itself or synthesize itself. It can move on into the S stage. Okay, so now cell division and cancer are related. So cancer is the result of unchecked cell division. Mechanisms that regulate the cell cycle just break down. They don't work anymore. The loss of control begins with a change in the DNA sequence of a gene that codes for one of the regulatory molecules. Faulty instructions will lead to a protein that does not function as it should, and any disruption of the monitoring system can allow other mistakes to be passed on to the daughter cells. So eventually all checkpoints become non-functional, and rapidly reproducing daughter cells crowd out normal cells, resulting in cancer, resulting in a tumor. And I've got a little bit of time, but I think I'm going to stop here before I go on to the next lecture, because this is a little complicated. But we will talk next about normal P53 and how that works versus mutated P53, because the gene that coded for P53 had a mutation, and what that actually leads to as far as the cell cycle and cancer.